I think we'll now make a start. Thank you very much all for being here, and Happy New Year. Today I'm very happy to introduce Simone Tiberi, who's an assistant professor in, I believe, the oldest university in the world, University of Bologna. And without any further delay, I'll let Simone give his talk.
Thank you. So thanks a lot for the invitation, for being here and online. I just add a further couple of comments about myself, more from a scientific point of view. I'm an applied statistician, I think very applied statistician. I develop in general in computational biology, and mainly I've done two things. I worked doing my PhD in systems biology, and I developed stochastic methods for single-cell fluorescent data. And then during my postdoc, so my PhD was here in England at Warwick. And then during my postdoc in Zurich, I worked in bioinformatics. But there, I didn't actually analyze data sets. I worked on developing methods, so developing statistical software tools, basically our packages for various omics data sets. And so this second part is what I will focus on today. So I'm going to give a brief introduction about transcriptomics, and then I will go through some of the methods we developed that do analysis on transcriptomics data. Okay, transcriptomics is indeed about transcription. And so you originally transcribed the DNA initially an immature or unspliced RNA, which has both exons, which are these colorful parts, as well as introns, which are these other intermediate parts. And then this RNA undergoes a second process that's called splicing, where the introns are basically removed and the exons stick together. That leads to mature or spliced mRNAs that are called transcripts. And then these transcripts translate into proteins. Additionally, this splicing process can actually happen in slightly different ways. And in most human genes, for instance, this is the case. And a single gene can actually lead to multiple mRNAs, so to multiple transcripts. And so this can actually lead to distinct proteins. And transcriptomics intuitively focuses on this layer here, on the abundance of these mRNA transcripts. So there's various types of data for transcriptomics. Today I'll focus on bulk and single-cell RNA-seq. On bulk data, you have an average signal across many cells. So you do not distinguish the signal that's coming from the various cells, but you actually have good resolution in a different direction because you have good resolution about transcripts. So you can actually distinguish the individual transcripts from the same gene. In single-cell data, you pretty much have the opposite. So you can distinguish individual cells, and this allows you to actually do separate types of inference on the cell types. But you have lower resolution on the transcripts. So it's quite hard typically to distinguish the individual transcripts. And so you collapse all of them at the gene level. And normally you do analysis only at the gene level. And then you also have spatial transcriptomics, which is like single cell, but you also have a spatial context, but we're not going to talk about that today. Now, there are various protocols, but generally, I think we can say that you have some RNA sequences, which are basically sequences of the transcripts, and you want to know what transcript they're coming from. So you normally align them to a reference, which can be a genome, but typically a transcriptome, and you basically look for what areas of this reference these reads are compatible with. So really, in terms of sequences, what sequences of your reference are compatible with these reads. And this step is called alignment. And after you've done that, you simply count how many reads are compatible with each entity you're studying. For instance, in this case, you have 7 reads here compatible with this, in this case, gene A, and 13 reads there compatible with the second gene B. And you do that for all your genes. And so in the end, you get what's called a count matrix, where you have genes or transcripts, exons, what you're studying on the rows. And on the columns, you get samples in bulk data, normally 6, 8 samples, or single cells in single cell data. And in that case, you have typically thousands of columns. And then in both cases, normally you start from this count matrix, although we'll see we also use a different kind of information. But you normally start from this count matrix, and you do your downstream analysis. And among the downstream analysis you can do, there are differential analysis, which is mostly what I focused on. Now, what are differential analysis? Well, differential analysis try to compare groups or experimental conditions, like healthy disease or treatments or tumor subtypes. And for each condition, you observe several samples or biological replicates. And the aim of these studies is to identify the genes or the transcripts that display some sort of difference between the conditions. Now, difference is a general word. It depends on what you're studying. The most classical thing to do is differential gene abundance. So you look for the overall counts at the gene level, and you see whether they are statistically different between conditions. But you could do the same on the overall abundance of transcripts or exons, or you could analyze other things like gene regulation or alternative splicing patterns. And the reason we do that is because changes in transcription can be involved in disease, for instance, if you're studying healthy versus disease, or can change depending on the treatment, or you can discover marker genes or marker transcripts, for instance, for cancers or tumor subtypes and so on. Now, obviously, for every gene or every transcript, every unit you're testing, you have a statistical test. And so that leads to thousands or even dozens of thousands of tests. And so typically you correct for multiple testing via Benjamini-Hochberg correction. So you then use the FDR to threshold significant and not significant results. So next I'm going to show you 3 methods that we developed to do different types of differential analysis from transcriptomics data. And all of them have been released as R packages in Bioconductor. Again, the aim here is not to analyze a particular data set. We didn't start from a particular data set. The aim is to release a method, to release a software tool that bioinformaticians, computational biologists can then use to do their analysis. So the first tool I'll talk about is called Distinct, and it's actually the only tool I developed in bioinformatics, which is not just for bioinformatics data sets. This is actually a general method that compares groups of distributions. But obviously, we thought of it in the context of omics data sets. And in particular, our idea was to develop a tool that could do differential gene expression analysis, so overall abundance at the gene level, we don't distinguish transcripts here, from single cell data. So single cell RNA-seq, but also in this case, other types of data like flow mass cytometry. Now, the motivation for the study is that there are a bunch of tools that do this kind of analysis, but the classical tools tend to focus on changes between means. And this actually works very well on bulk data, but on single cell data, you can also have more subtle variations that do not necessarily involve the mean, but they can still be biologically interesting. For instance, you can stimulate cells and maybe some cells are upregulated and others are downregulated, but the overall mean doesn't change much. Or you can have differences in the variance, leaving the mean roughly stable. And so these kind of methods struggle to identify those changes. And what we propose here is a full distribution method that is non-parametric and does not look at changes in the means, but it looks at changes between distributions. And so it can identify changes regardless of whether the mean is involved. So here we have a schematic of 5 possible types we conceived of differential abundance between red and blue groups. So in this case, this is the classical shift of the entire distribution. These are more complex bimodal changes. You see all 3 at the beginning involve a change in the mean between groups. And then you have more extreme cases where you have, say, a unimodal versus a bimodal, or 2 unimodal distributions with a difference in the variance. And these 2 distributions, these 2 changes do not involve a change in the mean. So what we do, so the first step of this thing is actually to look at, instead of densities, to look at the cumulative density functions that refer to those densities. Because there are always some areas of the CDFs that differ between conditions, even in these more extreme examples like here. Now, if we look at exactly the same plot, but now adding biological replicates, we have, say, 4 red and 4 blue curves. What we would like to do is to numerically quantify the distance we observe here, basically this area, between the 2 groups of curves. And the way we did that was to build a fine grid of several cutoffs. By default, we use 25, but you can specify more if you want. And then at each cutoff, we compute the average of the CDFs in one group, the average in the other group, and we take the absolute difference. And then we just add all these absolute differences, And that gives us a test statistic that should summarize how distinct the two groups are. And intuitively, this is proportional to the area you have between the groups. Now, obviously, here there is no symptotic theory helping us in terms of statistical testing and suggesting a null distribution. And so we use a permutation approach. But again, we had another challenge because in biological data, you normally have very few samples. So in this context, you normally have 6, 8 samples. So these are not enough to perform a permutation test. And so what we decided to do instead, since every sample has multiple observations, like every sample has hundreds or typically thousands of single cell measurements, we decided to permute the individual observations. And so we actually permute the single cell samples, the single cell measurements. Now we know that this violates the exchangeability assumption of permutation test because obviously the single cell measurements between different samples technically not exchangeable. And so this could lead potentially to an inflation of false positives if there is a large degree of sample-to-sample variability. But in practice, we've done several benchmarks, and this doesn't seem to be an issue in the kind of data that we analyze. Probably also because the data is always normalized before being provided to the test. And the normalization makes the samples a lot more homogeneous, and so it decreases the variability between replicates. And we do a similar thing if there are covariates like batch effects. We regress them out with a linear mixed model before actually applying the method. Now, I said we have single cell data, we can distinguish cell types, but I haven't used that piece of information yet. So what we normally do is actually to, cluster cells, typically in cell types, but you could have any kind of cluster. And then in each cluster or cell type, we perform the differential analysis as I described before. So what we get is differential gene expression that is specific for a particular cell type. So because some genes obviously could be differential in some cell types and not in others. And this is actually called differential state analysis in the field, but it's basically cell type specific differential expression. We've done several benchmarks. This is the main one here. We've done basically a simulation from a tool that allows to simulate single cell RNA-seq counts directly. So we directly simulate counts. And the way this works is that we basically start from a real data set. We infer the parameters from the data set, and then we use these parameters to simulate new counts. We simulate 2 groups of 4 samples each, and then we assume that 10% of the genes have a difference between the 2 groups, and so we simulate a difference, and for the other 90% of the genes, there is no difference in abundance. Now, in order to simulate and investigate performance in all the profiles I've shown before, we simulate from each one of the 5 differential profiles here. Okay, so we get a big picture of how the methods perform in each situation. And we consider a bunch of competitors. So first of all, the classical competitors are these pseudo-bulk approaches, which are the methods that work well on bulk data and that target changes between the means. So classical things like EJA and LIMMA. But we also consider mixed models and other non-parametric approaches like Kolmogorov-Smirnov test and also another permutation test. And also we investigated how robust the methods are to the normalizations. As I said, we have to normalize data, but there's various ways you can normalize data. And so we consider 5 different normalizations of the input data. So here, a bit confusing, I think, but these are kind of the key results. I'll slowly explain what's in the plot. So here we have the true positive rate versus its false discovery rate. So in each graph, we want to be ideally on the top left corner, high true discoveries and few false discoveries. And then for each method, you see 4 circles, squares, or still shapes. And so what we ask the method is to provide results for theoretical thresholds of the FDR, which are 1, 5, 10, and 20%. So we ask the method to retrieve results for these FDR thresholds. And these circles indicate the actual true positive rates and false discovery rates of the results that are retrieved from the methods. So obviously, we would like the actual false discovery rate to be as close as possible to the theoretical one. So we would like these circles or shapes to be as close as possible to the vertical lines. And you see sometimes they're not, and in this case, there is a strong inflation of false positives. In this method, for instance, we ask for a 1% FDR, but we actually get a 50% FDR. Second thing to note, there's a bunch of methods because we have 5 normalizations for every method, roughly. But you see distinct is in reddish, And then the pseudo-bulk competitors are in blue and green. Then there's mixed methods in gray, and the other non-parametric approaches in being violet. So first thing to note is that these approaches they have a very low statistical power in our benchmarks. And that is actually coherent with what was found in other scenarios, in other papers. So you see they're mostly in this region. And I think without making it too long, the take-home message here is that roughly here at the top, you see there's a cluster of methods, which are basically those from distinct as well as those from the pseudo-bulk methods. Because these three on the top are the differential profiles that have involved a change in the mean. So roughly, we do as well as the classical methods that target changes in the mean when there is a change in the mean. And then when you look at the bottom, so the more probably the rarer but more subtle differences that do not involve a change in the mean, you see that our methods are kind of in this region, reddish, while the other pseudo-bulk methods are more towards the bottom. And this is expectable because we have a full distribution test while the others target a change in the mean. And so we get a bit of an advantage in these more subtle variations between conditions. Now, in these examples, in these simulations, you see we don't get an inflation of false positives because our kind of actual FDR is always calibrated. It's always actually on the left compared to the theoretical thresholds. But we also investigated the false positives on real data. So to do that, we took some real data sets, and basically we had several replicates that belong to the same experimental condition, and we randomly separated them in two groups 3 times. And so this created artificial data sets where we actually don't have a real difference between groups, and so we expect p-values to be uniform. So we've done that 3 times for 2 data sets. And so here it's the result. See, on the top is the first data set, and the bottom, the second one. Now you see this thing is here in red, and then you have here some pseudo-bulk methods, which are typically known for having a good false positive calibration. You see that there is indeed a bit of an inflation of false positives because p-values are a bit inflated towards 0. But of course I'm biased, but I think it's quite a minor effect. It's not a major effect, given that this is real data, and real data is always very noisy. And this is also quite compatible with what we observe also for other methods which we know to normally control the false positive rates. Computationally, how do we do? Well, we're kind of average in the sense that we're here in the middle. The method takes about like 3-4 minutes on a laptop on a small data set. It takes a few more minutes on a bigger data set. We obviously take a lot longer than classical approaches, which are parametric, and they just look at the differences in the means because they're much faster and they only require a few seconds. But we take much less than other competitors that do permutation tests or mixed models. And one of the computational features of the method is that although it's in our package, we've coded the method in C++, and we run it in parallel, so we managed to bring down the computational cost to what I think is actually quite reasonable. Okay, so wrapping up a little bit this first part. So this method is to compare, in general, groups of distributions by looking at the CDFs. Although obviously we think of it in the context of single-cell data. We look for any kind of complex changes, whether they involve the mean or not. And I've shown this method on single-cell data, but on single-cell RNA-seq data, but we also use it on single-cell mass cytometry data. And it can in general be used on other types of another data sets because it just compares groups of distributions. And we allow for replicates and try to correct for covariates by regressing them out. And I think, well, I've wrote it on both pros and cons, but I think it's relatively computationally efficient given that it's doing thousands of permutation tests. But of course, bioinformaticians are demanding. And so for them, it's relatively slow compared to methods that take very few seconds. And we have a potential inflation of false positives that doesn't seem extreme, but it potentially is there. Okay, next I'm going to talk about actually the first method I did in bioinformatics, which is called bandits. Here we're changing type of data. Here we're looking at bulk RNA-seq data, so average signal across many cells, but higher resolution on transcripts. And here we take advantage of this higher resolution on transcripts because we actually study differences in splicing patterns. So if you remember, seeing that an unspliced mRNA, again, both exons and introns, can be spliced in different ways via a process that's called alternate splicing. So one gene can have multiple transcripts. Now, what we study here are differences in these alternate splicing structures. So what we mean in practice is that we don't look at the abundance, the overall abundance of genes or transcripts, but we look at the relative abundance within a gene. So I'll make a very simple example. If a gene says has 100 counts, 70 from the first transcript, 20 from the second, and 10 from the third, in differential gene expression, you would look at how those 100 counts change between conditions. Here, we look at the relative abundance within genes, so 0.7, 0.2, 0.1, and how these vector frequencies changes between conditions. And now, clearly, to do that, we would ideally like to have the abundance of the transcripts, so the 70, 20, 10 that I mentioned. But in practice, there is an additional issue that limits us, and it's a technical issue that's called multi-mapping. So it's very hard to observe those numbers exactly, because typically, if you remember the alignment step, we have those reads, and we don't know what transcripts they're coming from, and we align them by looking at what basically transcripts those sequences are compatible with. Now, if you do that at the gene level, typically there is very little uncertainty. So most reads map to one gene. But if you do that at the transcript level, typically there is a lot of uncertainty. So most of the reads are actually compatible with multiple transcripts. You don't really know which one they're coming from. So here, if you take a toy example with two transcripts, say blue and red, you see that these are RNA-seq reads. You see that some uniquely align to one of the two transcripts. And so you can confidently assign this one to the blue transcript and these ones to the red one. But these 7 reads, you actually don't know what transcript they're coming from. Basically, we have information vertically, but we would like to have it horizontally, which is basically what corresponds to the actual biology. So you can solve the problem and estimate counts at the transcript level. There are EM algorithms that are quite accurate at doing that. But the problem is that you have a lot of uncertainty in those estimates. And so you need to account for that. So instead, what we did was to take care of this assignment on our own. And so we input what's called in the field equivalence classes. So simply what transcripts each read is compatible with. In this, again, toy example, we will basically have that one read is compatible with the blue transcript only, 2 reads with the red one only, and then 7 with both blue and red here. And then we use a latent variable approach to allocate these multi-mapping reads to the transcript of origin. And so we do that within a Bayesian framework. Now, how about the gene level counts? How do they distribute once you've done this latent variable allocation? How do they distribute to the transcripts? Well, take a single gene in a single experimental condition. Now, we said genes have different number of transcripts. And so we use general K number of transcripts for the gene, and we have observed N biological replicates for a condition. So we assume that the gene level counts for a specific sample are distributed to the K transcripts according to a multinomial with some probability vector pi that basically reflects the probability that a read comes from a transcript. And we allow this pi to vary from sample to sample because of biological variability. But obviously, we also have sharing of information with a Bayesian hierarchical model, and so we have a common Dirichlet prior for the pi's. And this delta here of the Dirichlet can be interpreted in 2 ways. So first of all, well, you have the summation of the deltas is called the precision parameter, and it indicates basically the degree of overdispersion or the sample-to-sample variability. And these normalized deltas are basically the group level mean relative abundance of the transcripts, basically averaged across the individual samples. And what we want to study is basically how this pi bar changes between conditions. Now, I've shown intuitively that we have sharing of information across samples with the hierarchical model, but we also have sharing of information across genes, because obviously we analyze thousands of genes together, and there are some things that we can learn from other genes, even though we analyze them individually. And so what we do is that, all this is, you can see the number of transcripts changes from gene to gene, but there is one parameter which is in common, it's this precision parameter. So what we do is to fit an initial model, which is not ours, and get estimates for these precision parameters. And we use those estimates to formulate an informative prior for the precision parameter. Now, this is basically an empirical based approach, but it's extremely mild because the estimates are obtained from thousands of genes, and then when you analyze each individual gene, it only contributed in basically in a negligible fraction to the prior. And then we use a metropolis within Gibbs approach to basically sample from the conditional distribution of the parameters. So we have the hyperparameters that follow metropolis sampler, and then the hierarchical that are basically follow Dirichlet Gibbs sampler, and the latent states that follow multinomial Gibbs sampler. So if you see, like, most of the metaparameters are actually sampled from a Gibbs sampler. Only very few parameters are not sampled from a Gibbs, which makes the MCMC actually quite smooth, even though we have many parameters. Now, although this is quite smooth, obviously we want to assess somehow that the chains have converged, that we have a good mixing, and this is pretty much impossible to do visually because we have thousands of genes, but also because this is a tool for users who are not familiar with Bayesian statistics. And so we use an automatic conversion test for every gene to make sure that the gene has converged. And if it hasn't, then we run a second chain twice as long. Now, this is basically how we fit the model for every gene in every experimental condition, which obviously leads to a significant computational challenge because we have in the end thousands or dozens of thousands of full MCMCs that we have to fit. And again, we use a similar strategy to these things. We code it in C++, although it's in R. We run it in parallel, and the average runtime per gene is below 1 second. Although obviously, when you add thousands of those overall, it takes 1 or 2 hours on a normal laptop, depending on the size of the data set, which for bioinformaticians is a very long time. But for me, I think it's quite reasonable time being a Bayesian. Now, an additional detail is about the length of the transcripts. Now, not all transcripts have the same length. And intuitively, if two transcripts are equally abundant, if one is twice as long, you will observe roughly twice as many RNA-seq reads. And so we try to account for the length of the transcripts and to go from the probability that a read comes from a transcript, which is what we studied before, to something that we're actually interested in, which is the probability of expressing the transcript in terms of biological unit, regardless of the length. And so to do the conversion quite intuitively, we just divide by the effective length of the transcripts and rescale so that probability is up to 1. And this is useful for two things. First of all, because when we test for differences in the proportion between conditions, we look at these new parameters, so the probability of actually expressing the transcript. And secondly, because when we do the latent variable allocation, we use this parameter to allocate reads to the transcript of origin. And so intuitively, if a transcript is compatible with if a read is compatible with two transcripts, J and K, we basically allocate it to the two transcripts according to their probabilities, pi Tj and pi Tk. Now, so far I showed you basically how to structure the model, get the parameters in each condition individually. But now what we're interested in is actually to do differential testing for the gene at the gene level, but also the transcript level. And so what we do here is to actually fit the model separately and then compare the relative abundances that we obtain at the group level. And the way we do that is to consider their difference. So imagine you have two groups A and B. We look at the difference between the relative abundances. And so we introduce this omega here. And if those difference, if the relative abundances are the same, there are no difference in alternate splicing. And so the omegas are going to be 0 for all the transcripts. If there are at least some differences in alternate splicing, these omegas is not going to be 0 for at least some transcripts. Now, in order to test for this system of hypotheses, we actually tried a few things, including base factors, but none of them worked very well. And so what we ended up with is something I think highly unusual in Bayesian statistics, which is to actually go back to the frequentist work. So here we approximate the posterior of omega. Again, omega is just the difference in these probability vectors with a multivariate normal. And here the mean and covariance of the normal can just be inferred from the posterior chains. And this approximation actually turned out to be quite accurate in all the cases we considered. And if you do that, then it's straightforward to use a multivariate wall test and obtain a p-value that you can then turn into an adjusted p-value for FDR correction. And this is for a gene level test where we consider the entire vector of probabilities. And we do the same for individual transcripts. So we consider individual pi-k probabilities, so individual differences, and we check if they are 0 or not. And in this case, we use a univariate normal approximation and a univariate wall test. Now, the reason we do this double testing at the gene and transcript level is because obviously biologists are, once they have identified that a gene has differences in alternative splicing, they're interested in knowing what are the individual transcripts that are affected. And so here we can actually provide a proper statistical test for the individual transcripts so that they can see the results at a double resolution. Again, we've done several simulation studies starting from real data. So we, again, use a real data set, infer the parameters, and resimulate reads. Now, in this case, we're interested in the mapping uncertainty specifically. So we simulate at the read level. So we simulate the actual RNA-seq reads before alignment. So we then realign them and obtain the equivalence classes that we need for the analysis so that we make sure that the actual data we use has mapping uncertainty. And again, we simulate 2 groups, 6 samples each, 10% have different differences, 90% don't, and we see if we can recover them. As you'll see, we have several competitors. Just the highlight is that what actually motivated us is known as these competitors accounts for both the mapping uncertainty and the sample-to-sample variability. So they account at most for one of the 2 factors. Again, same plot as before, I'll make it brief. Bandits, we have a couple of versions. I didn't explain the difference. There were a couple of versions in green, and you see that Bandits is doing quite well because it's here. So it's just for given FTR, it has a good statistical power. And this is for a gene level test, and we checked exactly the same for a transcript level test, and we get similar results. Again, you see we have fewer methods here, fewer competitors, because not all of them allow for this double testing at the gene and transcript level. In this case, we actually were quite lucky because there was a real data set available in the literature where a good bunch of genes, 82 genes, have been validated in the lab to display differences in alternative splicing. And so we used this, and we basically assumed, a bit in an approximative way, that those 82 genes are truly differential and that any other gene is not. Of course, it's rough, but this allowed us to build something like a rock curve. And again, Bandits is here. You can see that we were interested in checking that these 82 genes were ranked towards the top, so that we're in the top, meaning with the smallest p-value or FTR. Computational, it's a similar story to distinct. We don't do terribly bad. Of course, I'm very biased here, but we do definitely, on the right, you see kind of the fastest methods. We're definitely slower than other competitors, because none of these other methods does a full MCMC approach with latent states. Even there's another Bayesian method, but it's actually an approximate Bayesian approach. But overall, looking at the bigger picture on the left, you see Bandits is actually here in the middle. So there are some methods which are slightly faster, but there are a lot of methods which are actually significantly slower. So I think, again, you have to wait a little bit, but it is, I think, a reasonable computational time. So wrapping up again this part, we analyze BAF data, we look for differences in alternative splicing, and we allow users, I didn't talk about that, but in the alignment step, you can align reads in several ways. You can do that to a reference transcriptome, genome, you can use several tools, and so we allow for a bit of flexibility. We allow for the alignment, both genome and transcriptome, and also to use a couple of the 2 most popular tools in the field. And we model the mapping uncertainty with latent states and the sample-to-sample variability with a hierarchical model and do the double testing at the gene and transcript level. We also have a correction for the transcript length, and I haven't spoken about that, but we actually see that it actually improves performance. So it's not just a theoretical thing. We see that it actually gives us a bit slightly better results. Obviously, this comes at a cost, mostly computational, but also in terms of flexibility, because our model does not allow for covariates like batch effects. Because I tried to include them, but in the end, it was not working very well, because I think it becomes a bit too complex. But to our defense, they are quite unlikely to affect relative abundances. Normally, batch effects covariates affect gene abundance, overall abundance, but we look at relative abundances within genes. It's quite unlikely that they have an impact. Okay, next I'm going to talk about a project which is not really completed, and it is a bit of an extension of Bandits. So this method is, again, working on single cell data, So again, we go back like distinct to single cell data. And it tries to extend the mathematical aspects of bandits to perform a different kind of analysis. So again, let's take the kind of splicing and transcription scheme of before. This is slightly different because it does not distinguish several transcripts. But again, you have DNA transcription, unspliced or immature molecule of mRNA, and then after splicing, you have a mature or spliced molecule. Now, I told you single-cell RNA-seq does not really allow you to distinguish the different transcripts, but it allows you to distinguish the spliced from the unspliced versions at the gene level. And this is quite useful because there are, for instance, there are several methods actually that study RNA velocity that is basically the derivative of the spliced mRNA. So you can infer or you can try to study what happens to the spliced abundance in the near future. And the assumption here is that if you have say a higher relative abundance of unspliced reads than you would expect at the equilibrium, then you will have in the near future, you will have a large amount of newly spliced mRNA, which is going to be larger than the degraded spliced mRNA. And so the spliced mRNA in the near future is going to increase. So you can think of the gene as being upregulated right now. Conversely, if you have a lower amount of unspliced mRNA than you would expect at equilibrium, the newly spliced mRNA will not fully compensate the degradation of the already spliced mRNA. So in the near future, spliced mRNA is going to go down, and you can think of it as being downregulated by now. Now, this is not something new, it's already in the literature. What we do here is to take this idea and try to use it to compare, again, experimental conditions. So we look for differences in the relative abundance of spliced and unspliced reads, and we speculate that if you have a higher relative abundance of unspliced reads in a condition compared to another one, then this condition is being upregulated compared to the other. Again, it's not absolute up or down regulation, it's relative between two conditions. And again, this is different from differential gene expression. In differential gene expression, also conceptually, you would look at the total abundance of spliced mRNA. Here we look at differences in the near future change, so in the direction of spliced mRNA. And again, since we have single cell data, just like in distinct, we first cluster them in groups, like cell types, and then each cell type, we aggregate the measurements, obtaining basically bulk measurements within a cell type, and we do the differential testing. So just as an example, you see in this case, in this particular gene, in this cell type, the relative abundance of unspliced mRNA is larger at 6 months compared to 3, and so we would speculate that 6-month brain organoid are being upregulated in this gene, in 6 months brain organoid is being upregulated compared to 3 months. Once again, you can estimate the abundance of spliced and unspliced reads, but there is a lot of uncertainty. And here, compared to what I showed before at the bulk level, uncertainty is actually at 2 levels. So we have uncertainty within genes between the spliced and unspliced versions. We call these ambiguous reads. And this is similar to the ambiguity we had before between transcripts. But we also have an additional layer of uncertainty, which is between different genes. So reads often are compatible between different genes in droplet single cell data. Well, this is quite rare in bulk RNA-seq data. And so the way we try to deal with these two sources of mapping uncertainty is actually different. For the gene level, we use a latent variable approach. So again, we sample the gene allocation of these reads. For the ambiguous reads between spliced and unspliced, that is actually quite hard to do. Because to do it, you need a good estimate of what is the probability that an ambiguous read is spliced or unspliced. But that is very hard to estimate because it depends on many factors that are not known. And so we still tried a latent variable approach, assuming that ambiguous reads had a 50-50 probability of being spliced and spliced. And it worked, but obviously it was not brilliant because that's a very rough guess. What worked best was actually to treat the ambiguous reads separately. So technically, you have a biological bivariate vector, spliced and unspliced reads, but then practically, you have a trivariate vector of data, which is spliced and spliced ambiguous. And so we work with this trivariate vector instead of the original one. And so we look then for differences in this trivariate vector. Again, if you take a gene, one experimental condition, we use again a Dirichlet multinomial parameter to allocate reads in a gene to the three conditions, splice and splice ambiguous. But additionally here, we have a second model, which is a multinomial model to allocate reads between different genes. And so this basically represents the total counts at the gene level across the three versions, splice and splice ambiguous. And a latent allocation in this case depends on both parameters. And indeed, for example, if you have a read that's compatible, say, the spliced version of gene G and the unspliced version of gene J, then you would allocate it to the first case with probability proportional to the probability of gene G times the probability of the spliced version of gene G. And the second case with probability proportional to the probability of gene J times the unspliced probability of gene J. Obviously, then renormalizing so that they are to 1. And again, we have sharing of information across genes with an informative prior, but in this case, we can go beyond what we did before, because before we only had an informative prior for the precision parameter. Here, you can see that actually all these deltas are in common across all genes, because all of them have exactly the three versions, spliced, spliced, ambiguous. And so we have an informative prior on all three Dirichlet parameters. And then again, we compare the average relative abundance with a multivariate wall test. So we have again generated a simulated data set. In this case, we actually don't simulate from parameters that are inferred from the data. We actually take a real data set. We take the counts that are taken from a real data set. So this is normally called a semi-simulated data set, because we actually take real counts from 4 samples, we split them into 2 groups, and then we artificially modify the counts in 10% of the genes to introduce a difference. But now these counts do not have mapping uncertainty because we're actually playing directly with the counts. And so we provide them again to another tool that's called MinNode that allows us to simulate from these counts at the read level. And then again, we have to realign and so on so that we have mapping uncertainty. And we compare against a couple of competitors that, again, this is what Tiberi does, don't account for this mapping uncertainty. And you can see that differential regulation is in green. Both of them suffer both the competitors suffer from inflation of false positives, but also from a very low statistical power. And actually, I didn't think about making the plot here, but these two competitors, if you take the same simulation before the mapping uncertainty, so before providing the counts to the read level simulator, they both work very well. So in principle, they would be able to identify the differences, but then when you introduce the mapping uncertainty, they really struggle to do that. So again, wrapping up this part, we have proposed this basically extension of bandits to study regulation between conditions. We try to account for these sources of uncertainty between genes and within genes, and for the sample-to-sample variability. Now, so far I showed the initial framework we developed, which is to do analysis at the gene level of single-cell data. So you can identify cell type specific changes, but only at the gene level. You don't distinguish the transcripts involved. Now we're actually extending this and we're benchmarking this on bulk data. So the idea there is to do the same kind of analysis on bulk data, so you don't distinguish cell types anymore. It's not cell type specific. But as I said before, on bulk data, you can actually go deeper at the transcript level. So we can actually study the individual transcripts which are being differentially regulated. And so this, I think, I like this idea because it would allow biologists to go deeper in different directions according to what they're interested in, either cell types or transcripts within a gene. Again, I didn't show a computational benchmark, but obviously the method is quite computational intensive, still reasonable, but definitely doesn't take a few seconds. And just like bandits, we don't account for covariates, but again, they're quite unlikely to have an impact on relative abundances. Normally they affect overall abundances. Now, lastly, I wanted to very briefly, not really advertise a position, I don't have funding, but advertise a potential application. So I have an ongoing project, which is integrating, it's a bit different from the other ones, it's integrating proteomics and transcriptomics. It was technically awarded a Marie Curie fellowship, which is a postdoc fellowship, but it's not compatible with my position. So if you don't know what it is, it's basically, the classical EU postdoc fellowship for 2 or 3 years. So I'm actually trying to recycle this and trying to look for someone who's willing to apply for it and do a postdoc with me in Bologna in the Department of Statistics. So if there's anyone interested, maybe online, or if you know anyone who would be interested, let them know, because the applications are in September, but of course I'd like to be contacted in advance. So let's get to the scientific aspect of the project. In this case, we try to do inference on proteins. Now, again, this typical thing in biology, we don't really observe things directly. We'd like to observe proteins, but we actually observe peptides. Now, peptides are usually compatible with multiple proteins. And this is very similar to the multi-mapping issue of RNA-seq, but this is to a much larger extent because you have a lot more noise in proteomics and because you have way fewer peptides than you have RNA-seq reads. So while with RNA-seq data, you still manage to do inference on transcripts, here it's almost impossible. And it's very difficult to do inference on the individual proteins at the isoform, or isoform level is the analogous of transcript level within a gene. So here, the classical strategy is to group proteins, typically do analysis at the gene level, but you don't really know what's happening within the gene on the individual isoforms. So our idea is to build a model and enhance the data that you have available and jointly model proteomics and transcriptomics data. Now, transcripts are a prerequisite and are correlated to proteins because mRNAs are then translated into proteins. So the idea is that you can enhance the information in the data by also looking at what's happening at the transcript level at higher resolution. And so jointly modeling both, the idea is to try and do inference on the proteins at the isoform level. And so what we would like to have is something that infers if a protein is present or absent, with a measure of uncertainty like a probability, infer its abundance, again with a measure of uncertainty like a credible interval, and then in a second stage, extend this so that it actually embeds multiple samples and you get a group level inference, and also do differential testing between groups, again at the isoform level. Just to give you a toy example, it's a potential real example that you haven't done. We know that a transcription factor is differentially abundant between melanoma subtypes, but we only have information at the gene level. So with this kind of tool, if successful, we could study, first of all, what are the individual isoforms that are present and their abundance, but also try to identify the actual individual isoforms of that gene, which are differentially abundant between subtypes. And one of the nice things about the project is that we're actually supported, this is a joint collaboration with a biological lab in the States, where they're doing experiments and they're helping us to test and benchmark the method. And we've done a preliminary model that was actually quite successful, but obviously there's most of the work is ahead. So if you're interested, let me know if you know someone. Okay, in terms of availability of what I showed, all the three packages I showed are available on Bioconductor. For the first two, there is a paper. The last one is actually not completed. We're still working on it. I think that in spring, we should have a preprint out. And lastly, I'd like to thank Mark, who has been my supervisor for 6 years in Zurich. All the people who contributed to the work as co-authors or with suggestions, particularly in the lab. And finally, just to conclude with an artistic picture of the David in Florence. Okay, thank you for your attention.
Why Florence and not Bologna?
Thank you very much, Simone, for this nice overview of your talk. There is time for questions both from the audience and online. So please raise your hand if you want to ask something. I think I'll make a start. So in your bandage model, you mentioned that you tried to add covariates, but that didn't work well. So can I ask how you tried to add the covariates in the bandage model?
We tried. So basically, these relative abundances, we modeled them as a function of the covariates. I think I'll look. don't know what function, it's something like a logistic function of the covariance. The thing is, it was not completely unsuccessful. But we've run a few simulations, even very extreme, where we simulated like batch effects, even very extreme batch effects. And the bandits model with covariates was working worse than the original model without covariates. I think that the issue is that the extended model with covariates is technically more accurate, but then you make the model more complicated. And so the additional complexity that you have in the model is, makes it worse than the benefit of actually having an accurate model. Because even in very extreme and completely unrealistic scenarios, it was working worst. And since I know that it's very unlikely that covariates affect relative abundances, we decided to not to release that version.
Mm-hmm. I see. Yeah. Perhaps in that case, with covariates, maybe a hierarchical model among. Instead of running a single model for each one of them, perhaps having a joint model for all of it. Of course, it would be very slow, but perhaps.
But it is a joint model, right? So we infer all the parameters together.
Yeah, but sharing information regarding the covariate effect.
Oh, no, but it was shared. It was shared.
Okay.
Yeah. I mean, just like here, you have, because the thing is also, when you have covariates, you don't have a given sample anymore. I think that was the key. Because here at the PI, it's about a dirichlet prior, which is conjugate, so it's very big to sample them. If you have covariates, you have a common prior with a hierarchical model, but it's not conjugated anymore, so you need to go with a metropolis or metropolis distinct sampler, and then things become very, very slow and mixing is worse.
So let me see if there's any question online. Please feel free to unmute. Another.
Yeah, I'm not a biologist, but I had a question that maybe a biologist would ask. When you're talking about distinct, you said you could test hypotheses other than those relating to the mean, which I thought pretty interesting, but what kind of biologically phrased hypotheses somebody could test by using those methods? Like, remember, like looking at differences in the distribution rather than the moments.
I think they're rare events. I think it's a minor improvement, okay, because most of the times you're interested in differences than mean. But there could be cases, as like the example I showed here, where you have a difference in the distributions that maybe it's biologically relevant, but it does not involve the mean. I think the classical thing is really a difference in the variance, right? It could still be indicative if you have a lot more variability between the cells in one condition than in the other. If you look at the means, you're not going to detect that. So those cases could potentially be interesting, and that it's definitely a sample of variation. It's not the main aim of differential analysis, but it's more of an additional thing that the classical tools are not going to detect.
Are there any kind of, I've done like differential expression analysis with RNA-seq before, but are there any kind of canonical differential variance tests, like F-test based?
Yeah, I mean, you could do other kinds of tests. I mean, also basics like from Catalina Vallejos, who was here, does differential variance testing. But this is more a way to put any kind of difference together, right? So also like differential variance, but in this case, maybe this case it's kind of a different way where you can think that some cells like are upregulated in one condition. Maybe you treat them and they're upregulated and others are downregulated because they behave very differently when you inject the treatment.
What does the DB stand for, sorry?
DB is like kind of unimodal versus a bimodal. And you could think of it, I think the classical example is like you treat cells and the treatment kind of stimulates and upregulates some cells and it downregulates other cells because it behaves in the opposite way. Obviously, this is kind of the perfect shape example. But if something similar happens and the mean is not exactly the same, but it's similar, you're not going to see that.
David,
so for your differential, I think differential regulation package, so you have your spliced, your unspliced, and your ambiguous. And I was just wondering how many of the reads can you actually identify as being in the spliced form? Because presumably you can only identify them as being spliced if they fall across an exon junction. Whereas,
yes.
Like, so do most of your reads essentially fall into unspliced or ambiguous, and really you're looking at the differences between the number of unspliced and ambiguous reads?
So I don't want to say something inaccurate, so I don't remember.
Okay.
But you can identify, as you say, if they jump over a junction, which is basically like here, okay? If they jump over a junction, right? Then you know they're just spliced. They're only big. I mean, you know that they're unspliced if they somehow touch even partially the entrance.
Yeah.
They're only spliced. I mean, they're ambiguous if they are perfectly on the exons, and they are spliced if they jump over the junctions. But I don't know how frequently that could happen, because if the exons are short, you could have lots of junctions, right? So you could potentially have many spliced reads. I feel like saying most are spliced, but I don't remember. So I'll skip the answer.
Can see John wants to ask a question.
Well, it's more of a comment. I was just following up the question about.
I don't currently hear you, so perhaps our volume is low.
Is that better?
We'll skip. Yeah.
So I think we have no volume. Go ahead.
We can now hear you, John, for sure.
Oh, okay. It's more of a comment. I was just following up the question about biological mechanisms that would generate changes, but not changes in the mean. And it struck me that if your grouping by cell type is maybe quite coarse, there will be variation within that grouping, and you could get cells doing completely opposite things, you know. So if you cluster by T cells, for instance, it could be one variety of T cell goes up and one variety of T cell goes down, or something quite complicated, or it could be your treatment changes the proportion of different subtypes that you have in that sample. So which you know, this might be much more sensitive to than a mean test would be. So I think you can come up with biological stories where this would be quite useful. But I'm not a biologist either, so you shouldn't put too much faith in that.
Thanks. I don't think I need to add anything, right? I think you replied better than me, the previous question.
So I think there are no more questions.
Was there also a question? I think your comment was really accurate. I don't know if you also kind of implied a question for me. No, probably not.
So we're out of time. So please join me in thanking Simone again.